Hello, my name is Robert Bennett, and I'm doing a presentation on Dr. Laura-Isobel McCall and her contributions to understanding Chagas disease. Treating parasite diseases remains a big challenge in medicine because there are many unknowns about the systemic effects parasites can have on their hosts. Dr. Laura-Isobel McCall has made key contributions in understanding causes and treatments for these neglected diseases. The person who sparked McCall's interest in infectious diseases was Louis Pasteur, and this interest inspired her to continue her education at the doctorate level. Her PhD was completed at McGill University in microbiology and immunology, focusing on the understanding of the parasitic disease Leishmaniasis. Her work at the University of Oklahoma is primarily focused on Chagas disease. Her process of using mass spectrometry-based metabolics to understand the distribution of the parasite metabolites and the areas that have been damaged within the host has been remarkably effective. This has helped to tell where the parasite prefers to cause havoc. Further experimentation has been done by taking cross-sections of heart tissue from the lab mice to see where in the vital organ the parasite prefers to attack and if these locations could be life-threatening. The most remarkable thing about her work is that she has expressed that her goal is to use 3D mapping to make a real-world impact and help develop therapies for diseases that have otherwise been ignored. Chagas disease was identified in 1909 by a Brazilian physician named Carlos Chagas. This disease is common in Mexico, Latin, and South America, primarily in poor and rural communities. In April 2019, the CDC estimated that more than 300,000 people in the U.S. have been infected with Trypanosoma cruzi, which cause Chagas disease. This infection is commonly contracted by coming in contact with the fecal matter of the kissing bug. It is a blood-sucking bug that feeds off human and animals. And below you can see on the left and right, this is a picture of the kissing bug, and in the middle it's a microscopic picture of the manifestation of the parasite within the body. The primary way a host passes Chagas disease to another human is transmission from a mother to child during birth, and this is considered congenital transmission. It can also be passed by blood transfusions and/or organ transplants. Where Chagas disease is common, countries have begun testing blood donations to rule out that type of transmission. Once transmission of the disease has taken place, symptoms similar to influenza A or B are almost immediately expressed in a host, and this is considered the acute stage. The acute stage of Chagas disease is the best time to try to treat and suppress the ability of the parasite to spread and/or reproduce throughout a host's body. The majority, 70 to 80% of hosts, will continue their lives as asymptomatic hosts, not showing any signs or symptoms. For another 20 to 30%, Chagas will have severe and sometimes life-threatening complications from the infection. The experiment described in the journal article was designed to monitor gastrointestinal infections by T. cruzi, the known cause of Chagas disease. The experimental sample size was 40. 20 mice were infected by injecting intraperitoneal T. cruzi culture that would emit orange and red light or bioluminescence to help with detecting concentration of gastrointestinal infection and area of concentration. The other 20 subjects were left uninfected with the parasite and given a placebo of amino acids, vitamins, and glucose to determine the differences of infected versus uninfected. As the mice were monitored, the lab technicians collected fecal samples and froze them with liquid nitrogen for later examination and comparison. Metabolites were analyzed by mass spectrometry, and sequencing was used to identify gut bacteria. This experiment was done to study the microenvironment of the gut microbiome and the metabolome and how it is affected by the parasite. The manifestation of the parasitic infection within the intestinal tract of a host or microbiome can cause nutrient uptake inhibiting for the host and parasite. This microbiome could also cause problems with antiparasitic immune response from the host's natural body responses. After separating fecal matter from both infected and non-infected mice from 21 and 90 days post-infection, this helped enhance the ability to determine the host's inflammatory responses to the parasitic disease. This experiment has been essential for education of Chagas disease and making progress in prognosis and treatment for people who suffer from it. After close examination, some of the microbiome characteristics, such as bile acid metabolism by gut microbiota similarities, were noticed with other gastrointestinal inflammatory diseases, such as Crohn's. The manifestation of Chagas disease has been shown to present itself within a gut parasite reservoir and can move to the cardiac area of a host, causing the severe to fatal Chagas disease complications. Here's a list of my sources. I hope you all enjoyed the presentation as I did in the research, and I am sorry I can't attend the Q&A, but I appreciate all your attention and interest in this